I want you all to envision a world where there's no world hunger, there's significantly less poverty, and the medical industry, the medical world as we know it, has changed completely. I want to welcome you all to my presentation to genetic engineering, and I hope by the end of this presentation you'll all walk away feeling inspired to look into this a little bit further. Now, I'm sure you're all wondering, what is genetic engineering? Well, it all starts with the fundamental piece of life, well, at least one of them. Do you want to guess? Take a stab at it? DNA, exactly. DNA is deoxyribonucleic acid. Now, as most of you probably know, DNA has a double helix structure like this. Like this. So you've got combinations of A's and T's and C's and G's. Now, well, from here, a larger complex would be the genome, which is just the full set of DNA within an organism. Now, genetic engineering is just the manipulation, the editing, and removal of certain genes within an organism. Anything that's living, I'm pretty sure in every scenario, has DNA. It's found in the nucleus of a cell, and that gives it its instructions on what to do, how to function. So, as I was saying, DNA is just the editing of genes. Now, there are multiple ways of genetically engineering an organism, but I'm going to be talking about the three most, not important, but maybe popular ways of genetically engineering things. So, 1, CRISPR, 2, TALENs, and 3, ZFMs. So to begin with, CRISPR. CRISPR is kind of like a biological immune system. In the case where you have a bacterium, say it's swimming in a lake, and in comes a virus, that virus's goal is to take over that cell, to replicate, and then disperse once again. In the case where a bacterium survives the attack, then it'll go and take that virus DNA, that viral DNA, and put it in its own DNA code in a system called CRISPR. It saves the DNA as something called RNA, an RNA copy, which is just in most cases, a single-stranded protein of DNA, forgive me. So, it'd be like that. Within a larger DNA code. So it'd be a little bit like that. So now, say that the virus comes along again, and once again, its goal is to replicate, take over the cell. Then the bacterium has a really, really unique and special way of protecting itself. It'll go and take this protein called Cas9, and it'll combine with RNA to make something called, well, CRISPR Cas9. And what this will do is that this will go out and seek the virus that has infected the bacterium, and once it finds it, once it has an exact copy, an exact match of the DNA found, then it'll go and it will cleave the DNA, but only one strand. So that would mean that it would only take out one piece, or one of the two strands, so one of the helixes. So it would be, instead of like this, it would be rather like that. So now you don't have a functional piece of DNA. They've just taken out a gene, making it, well, sort of immobilized. So this is incredibly important because what scientists have been able to do with this is they're able to genetically modify their own organisms using this system. They can make their own RNA, combine it with the Cas9 protein found in bacterium, the CRISPR system, and what they can do is that they can genetically modify organisms with incredible precision, which is awesome because this is one of the most recent ways to genetically engineer. So the previous ones were not as accurate. You had double helix breaks where the organism had to repair itself, and sometimes, I think a while back they were using radiation to find random genetic modifications, which was not specific whatsoever. So, moving on, we have a brief history, of course. So, in 1953, we found out that the DNA was made of a double helix structure by Francis Crick and James Watson. Moving on to 2012, you had the discovery of the CRISPR system by Jennifer Doudna and Emmanuel Charpentier. They both won Nobel Prizes. And now we have the future. Anything starting from this point onwards has incredible, it's unimaginable what could come of this, because there are so many opportunities, so many ways to change the world that it's just unpredictable. So, why CRISPR? Like I was saying, it has incredible precision. You can target a specific gene within an entire genome with CRISPR, making it so powerful that it's just revolutionary. Furthermore, it's cheap. It's significantly cheaper somehow than other ways to genetically modify organisms. And finally, it's accessible. You can transfer this, even GMOs, genetically modified organisms, once they're made, all around the world. Sorry, do you mind if you just move your chair a little bit? You can transport this all around the world, potentially. It really could be very useful. Doing this, well, it can, like I was saying, solve a variety of problems. We'll come to this again at the end. Then you've got ZFNs. So ZFNs stand for zinc, finger, nuclei, I believe. I'm not sure about that. It's either nucleus or nuclei. Plural of nucleus. Is it?
Yeah.
Nuclei, perfect. So these are made up of two parts. You've got A, zinc fingers, or zinc finger. We'll forget about that. Nuclei. So these two bits when combined, have an ability to genetically engineer organisms. So you've got zinc fingers that come, I read, funnily enough, comes from the African clawed toad, which is awesome. So I'm just going to go with that, African clawed toad. Interesting, but, well, it's science. Then you have nuclei. So nuclei contain DNA, right? So when you combine these two, these have the ability to cleave DNA, and this has sort of kind of like a function of RNA, where it shows you where to go and what DNA to cut, almost specifically. When you use ZFNs, they create a double helix break as opposed to one. So it cuts out an entire gene, doesn't just disable it. So then the organism has an ability to grow back and repair itself if it's not completely destroyed in the natural environments. So those are ZFNs. Moving on to talons. Talons, once again, made up of two parts. One, well, tails. And another one, nuclei once again. Same thing as last time. Nuclei have the ability to show the editing part of the system, where to go. It shows where to go, what to do, what exactly to cut. And then tails are once again found in bacteria, naturally. It's a naturally occurring protein. So when a bacteria wants to go ahead and infect a plant, then during that process, when it's during the infection, then it'll have tails to go ahead and cleave DNA once again. So super cool stuff. The only problem is any drawbacks. So, because this has been found so recently, especially CRISPR, there's no way to tell what would happen if we were to genetically engineer things on a massive scale. It's incredibly risky to do it on ourselves, and even the consumption of genetically modified organisms, whether it be plants or animals, there's no way to tell what it will do to us. Then, second point, something that I like to call designer baby phobia. The analogy is that in a world where we genetically engineer ourselves, parents will be able to pick what height, what eye color, what hair color, anything really about their children would be. So, as you can probably think right off the bat, this is kind of problematic because, well, you don't have the morality to genetically engineer the kids. You don't have the ethical right, so it's not moral. But B, there's a sort of like a political side to this, because if you want to genetically engineer your child before they're born, then, well, say that you engineer them to make them stronger, taller, then they have a better probability of success in life, meaning that it might be slightly unfair to the people that are not genetically modified. So what accessibility would they have, these genetically modified children, compared to non-genetically modified children? I mean, you could even have these kids, once they're older, taking power over others. So you could have a sort of, even like a dystopian society. So it's a scary thought to think of, but I think that if we are to move forward, I think it's incredibly important that we keep a close eye on these things, and that we always take into account ethics and, well, what the right thing is to do, always. And like I was saying, morality, we do not have the morality to genetically engineer children before they're born. So if, in extreme cases, say that you have a child that's born and they're like, hold on, why are my arms on my back? And what are my ears where my feet should be? I mean, that's just not, doesn't work. It's not moral. So even if they have something positive coming out of it, they do not have the choice, which is just not right. So, there's actually a real world case of this by a man called He Jiankui. He was the first man to genetically engineer humans. So there were 2 twin girls while they were still being developed in the womb. He successfully modified the 2 twin girls to be resistant to HIV, which is a significant scientific breakthrough. However, like I was saying before, he had no consent. He had falsified regulatory work, meaning that it was illegal what he was doing. He didn't get the paperwork done. And then, I don't think that the world is ready. We still need time to tell what genetically modifying people will do. Like I was saying before, even the consumption of GMOs, there's no outcome that's foreseeable in the near future. So I think that people are scared of stepping into the new reality. We have the technology, we really push it, and we really want to get things done, then it's possible. But I think that it's very important that before we do this, that we have a firm grip on the rules and making sure that everybody understands. Because right now, this guy unfortunately did not follow the rules, and he was punished. I'm pretty sure he was fined something like £500,000, and then he lost his license, and he spent 2 years in jail. So it can be really quite severe. But on the bright side, what are the solutions, the potential solutions to this? There are countless solutions. That's the answer. Anything that has a problem with the genes can be solved. So anything that you see on the screen, I'm going to highlight cancer because that's probably one of the extremely important ones. Has the ability to be solved. With the right amount of research being done and the right amount of investments and commitment, then it's possible for it to be done within well, I'd say 2 to 5 years if we really, really pushed it. But it would be tough to even test this. Say that you have a gene, because it doesn't even need to be curing diseases. Say that you have a gene that you don't really want. Let's just, for simplicity's sake, we're just going to say that it's you're still unborn, so you'll still be in the womb. Let's say, bringing this back to the designer baby scenario, your parents want to genetically modify you. If you have, say, something like Down syndrome, then, although, you know what, let's go back on that. I don't think that's actually proven, but say that you have something like sickle cell anemia. You have sickle cell anemia, and you want to get rid of it. All they would have to do is that they would take you to a lab, and then with a little sort of injection, then you'd be cured. That would potentially save your life. It could give you a far better opportunity for success in the future, and you could have a better life. But as we were saying before, it can be very difficult ethically. So I've actually done some work in this, and I've managed to publish an article on Medium where I was talking about the utilization of genetic engineering to cure Alzheimer's disease. So for those of you who don't know, Alzheimer's disease is a neurodegenerative disease that causes a significant loss of memory over a prolonged period of time. My grandfather actually has this disease, so I was sort of inspired to take a closer look into it and actually see what solutions were being developed today. So I was talking about this here, and of course, talking about what the three were, the three genetic engineering techniques were. Then going on to the first one, the University of California, San Diego. They are, let me see if I got this right. Yes, they are using CRISPR to tackle the production of something called beta amyloid. Beta amyloid, that's the abbreviation. Beta amyloid comes from something called amyloid precursor protein, APP, which is right there, APP. So they're using CRISPR to make sure that there's either, well, in a normal scenario, from APP you can either get A, beta amyloid, or B, a more protective form of beta amyloid. So what they're doing is that they're making sure that you get this one as opposed to this one, because this one is bad. It's thought to cause Alzheimer's, and this one, it's good. So moving on to the next one, you've got a company called BioMed. BioMed is working on something called, well, it's actually a drug. It's actually got a weird name, BIO080, something of the sort. But what they're doing is that they are trying to, let me see if I've got this one right again. They are working on a gene silencing drug to prevent the form of the tau protein, which comes from something called the MAPT gene. So this is known to cause Alzheimer's disease. By the way, it's worth noting that I'm referencing Alzheimer's as AD. So if you see that, just to point that out. So since we know that this is causing Alzheimer's disease, then it's good because we're using a really effective way of genetically engineering things, and we are preventing the development of a protein coming from a gene. So we're sort of like preventing the problem at the origin. So then finally we have UltraGen YX. These guys are once again targeting beta amyloid, but instead, as opposed to the University of California, San Diego, they are forgive me just a moment. These guys are, of course, because beta amyloid causes plaque to be formed in the brain, which is what's thought to cause Alzheimer's disease. These guys are actually tackling it with a drug, just like BioMed, but they've got a completely different approach as opposed to BioMed. as opposed to the University of California, San Diego. So, of course, I was talking about Hee Jung Kui. So from there, we can move on to my second piece of work, which is a video that I published on YouTube on this website called Benchling. And Benchling is a way to run different experiments based on genetic engineering that show you what the outcomes are of various experiments. Well, anyway, the point of it was that I was doing something called a restriction enzyme digest. Now, what this is, or rather, we'll start with what a restriction enzyme is. A restriction enzyme is an enzyme found in bacteria that recognizes certain signatures. So, in bacterium, you have something called methylation, which is just a signature on certain DNA saying, this is me, and this DNA that's entered my body is not me. So what the restriction enzymes do is that they see which one's which, and they attack the DNA which is not a part of it. So what I was doing is that I was extracting a gene from a plasmid, which is a circular piece of DNA, and what I did is that I ran the experiment to take out the gene. So at the end we had a result of the experiment. It was like a digital copy of it. So you had one part of the gene that was on one side and you had another part of the gene that was on the other side. And we managed to take out this gene from the plasmid, the PCBF1B TTIO. We managed to take out a gene called the thermus thermophilus, which from what I gathered stands for TTIOs was, forget about that. But it was really cool because I managed to get some experience from this and it turned out going really well. So then, moving on from there, I wanted to touch upon the popular GMO, the flavor saver tomato. This was put into US markets in 1994. And what it was, well, a tomato. What was special about this tomato, by the way, it is the one on the left side, not the right side. That's a regular tomato. This is the flavor saver tomato. It is a genetically modified tomato, so it's a GMO, but it is designed to stay on the shelves for way longer. It reportedly has a better taste, which I don't think was the actual intention, but well, and yeah, it works better as opposed to a regular tomato. These ones are way more efficient at being held. Once you buy them, they can stay on your shelves for, I'm pretty sure it's like a month or more, so it's crazy. At the beginning I was talking about a world with no hunger, a world where everybody's fed. This was the beginning of an attempt at that. The only problem was, like I've mentioned before, that nobody knows what will happen if you consume GMO. So it was tried a few times, nobody's actually gotten sick, but it was not reported, and there's no way of telling what the effects were. So until we're saying another half century, I would say, nobody's actually going to properly trust these. So it's still available to be purchased, I'm pretty sure, in some places, but nobody actually trusts it. So I want to thank you for watching this presentation, and I hope that
Thank you very much, Adria. Lots of interesting detail in there. A lot of what you touched on is relevant to A-level biologists who will be studying CRISPR-Cas9 and restriction enzyme digests. Does anybody have some questions for Adria? I'm sure you're itching to ask something about the future of genetic engineering.
Yeah, if you guys just want to go back to a slide, I can explain something a little bit more. Please go ahead.
Where would you say the line between preventing a disease and having a designer baby is like morally acceptable?
That's a good question. It's actually a very tough question to ask because no proper answer. I think that there should be a line drawn where it's either life-saving or it's just for cosmetic purposes. If you're going to save a child's life, then that should be a priority, as opposed to somebody wanting to change the height of their child. Cameron?
But what about if we could genetically modify babies to have increased lung capacity or
That's fair.
resistance to cancer cells?
Then that wouldn't exactly be cosmetic, but like I've already mentioned, it's problematic because they can get a better chance of life. They can get an increased chance at being successful. So that might not be able to happen. It would be nice. I know that you row, so do I. It would be nice, but it might not be able to be the case. Quint?
You know the guy that went to jail because he genetically modified his
Yes.
twins? Why did he go to jail? Because he helped them, no?
Well,
Can't,
that's
can't
arguable.
be.
arguably he did, but I'll just go back to the slide. He did not have the children's consent, so it was an ethical problem. B, he had falsified regulatory work, so it was illegal. That was the bottom line. But in general, even if he falsified, even if he actually had the right paperwork, then he would have still been challenged because it just was not seen as right in the eyes of the world.
How is genetic engineering regulated in a way that prevents dangerous and inappropriate uses, but also has the regulation team or boards themselves aren't being fair and just, not letting their own biases?
Yeah, that's very fair. I would think that anybody that wants to genetically engineer something would have to have a license, and then there would have to be, I'm not sure, I'll have to go back and check this later, but I would think there would be like regular checkups on what they're doing. You have to publicly state what you want to do, what your goal is. But I'm not sure, I'll have to get back to you on that one.
Would you trust any genetically modified food today?
I think I'd eat a flavor saver tomato. They're better. They've got a better taste. Yeah. From what I've seen, it's just changing a gene, and the gene is just a bit of protein. Then, well, I don't see a problem with it, but it's definitely up for debate.
Where do you see the future of genetic technology now?
Well, like I said at the start, it has incredible potential. It can branch into any industry, really. You could genetically engineer crops to have more yield. You could make trees give out more oxygen. You could, it could go anything from the medical industry to the food industry to even the atmosphere.
Yeah.
So it can go anywhere, really. Yep.
Do you think that genetic engineering could replace jobs in the future, in an agricultural context or medical context?
I don't think so. I just think that it would be a smaller pool, and therefore more competition. Because if you have more yield, then you have more product to give. Therefore, you have less pressure on demand. So, to answer your question, I don't think it would replace jobs. I just think that it would make it harder to do your job in the agricultural industry, at least. In the medical industry, I think it would just improve everybody's life just a little bit more.
Say for example, a crop suddenly needed to be watered far less and had way lower maintenance, well surely.
Exactly. Well, if it has lower maintenance, you still need to have the people to actually take the crop and then sell it. It would have to go through the supply chain, so you'd still have jobs. Maybe it would just decrease the amount of jobs. So yeah, I mean, maybe it would. It wouldn't necessarily replace their jobs. It would just minimize make it harder to do.
I'm not sure if they're genetically modified, but I've read that strawberries nowadays, they're bigger and healthier in a way.
Yeah.
Do you think because that's working strawberries, it means other fruits will also have a bright future?
I actually saw this a while back on TikTok. when we first found strawberries and started selling them on a massive scale, they were a little bit more wild, so they had a different color. They were a lot paler. They didn't have such a delicious red color. They were smaller as well. But as they become more sort of popular in urban areas, and they've been distributed more widely, and they've been given more water, and they've been able to reproduce on massive scale, because strawberries are really popular because they taste great. It's the genes have changed slightly. It might be the case that they did genetically modify some of them, but I'm not sure. The normal red strawberry as we know it probably just was environmental cost. Anything else?
What might you do before you take the step to genetically modify? How have we come to have such amazing strawberries and strong wheat? What do we do? You know, I'm sure some people are. Any ideas? Yeah.
Is that you would keep producing a particular type of crop or and you keep using seeds from that until you have a special breed or
Yeah.
I think
So
putting it under less stress environments.
Yeah, and selective breeding. Yeah.
Yeah, I was going to say selective breeding, same companies.
Absolutely.
You've got rapid evolution.
Do you think regulatory bodies should distinguish between things that are genetically modified through selective breeding means and synthetic means, or as you discussed, or we should?
To put it in short, probably, but I'll have to get back to you on that one again. I'll have to think about this a little bit more.
You've had some fantastic questions, which is really good. It's made you think, it's made everybody think, which is that's what this is all about, which is brilliant. So thank you so much for your attention and for your questions. Come and have a chat if you get anything else you want to ask Adria, and please make sure you've signed the register, and I look forward to seeing you all next week as well for our next chat.